The protein product of UL56, referred to as PUL56, is a large subunit of a viral terminase that cleaves viral DNA after it has been replicated so that it can be packaged into new viruses. It has been found that mutations in UL56 can either increase or decrease levels of antiviral resistance. Those viruses that are drug resistant are especially dangerous. The first step in this process was to create a mutated UL56 gene. To do this, we designed a mutated primer that could be used for PCR in order to create several copies of the mutated gene. We ordered both a forward and a reverse primer, which were both used for PCR. This results in a forward and a reverse product. Next, we used homologous recombination to insert the designed gene into a bacterial plasmid. In order to increase the chances that homologous recombination would occur, the plasmid was cut in two places so that it was linear. Then the linear plasmid was added to the primer products with a recombinase enzyme, which would carry out the reaction. This resulted in a circular plasmid that contained the mutated UL56 genes and a gene called mCherry that would allow us to visualize the protein product. mCherry fluoresces a red color so that a protein's activity can be visualized. The mutated plasmid was then incubated with stellar cells and then shocked to encourage transfection, in which the cell takes in the plasmid. Next, it was time to eliminate the stellar cells that did not take up the plasmid. The plasmid contains a gene for antibiotic resistance. The cells were grown on an agar that contained that antibiotic. This antibiotic killed all of the cells that did not contain the plasmid. We were left with only stellar cells containing the plasmid. Next, we needed to figure out what effect the mutation had on the protein, if any. In order to do this, the stellar cells were given another plasmid called a reporter plasmid. When PUL56 is present, it will bind the A site, blocking the transcription of CFP by physically blocking the RNA polymerase. The amount of red and blue fluorescence would need to be measured on 4 types of cells. First, cells containing only UL56. Next, cells containing only CFP. Third, cells containing normal UL56 gene and CFP. Lastly, cells containing the mutated UL56 and CFP. The first type of cells should only fluoresce red. The second type should only fluoresce blue. The third type should fluoresce the same amount of red as the first and less blue than the second. The fourth type of cells should fluoresce the same amount of red as the first and third. The amount of blue is what provides information. If the fourth type of cell has less blue fluorescence than the third type, the mutated PUL56 has a higher affinity for the A site than the wild type. If it has more blue fluorescence than the third type, the mutated PUL56 has a lower affinity for the A site than the wild type. If it has the same amount of blue fluorescence as the third type, then the mutation does not affect affinity for the A site.